The overall goal of this procedure is to measure somatosensory processing of light touch in young children. This is accomplished by first setting up event-related potential methodology and an aerotactile delivery system. The second step is to place an EEG net onto the child in order to measure neural activation in the brain. Next, the stimulus control application programmed with an airpuff versus sham paradigm is run. The final step is to process the data acquired from the event-related potential trials. Ultimately, by comparing the response to real tactile stimuli and to the sham, one can show cortical activation in response to light touch.
So the main advantage of this method compared to some other methods like MRI or electrical nerve stimulation is that it's very child-friendly. It's fast, it's painless, and it does not require active participation, which allows us to do it both in infants and in young children.
To program the stimulus control application for this protocol, first set up two serial commands. One command should identify the puff, the other the sham. Have the stimulus control application send the commands to a microcontroller. Have the microcontroller generate a 20 millisecond duration TTL pulse to the corresponding digital output channel. This output should be split into two lines, one for the digital input to the EEG recording system